The SARS-CoV-2 virus initiates infection using the trimeric spike glycoprotein. This protein is the molecular key to cell entry. The spike protein is composed of two functional subunits, S1 and S2. The S1 subunit contains the receptor binding domain, or RBD. The RBD specifically targets and binds to the angiotensin-converting enzyme 2 receptor, known as ACE2, which is present on the surface of host cells. This binding event is critical. It triggers a significant conformational change within the spike protein structure. Subsequently, the S2 subunit executes the membrane fusion process. This fusion merges the viral envelope directly with the host cell membrane, allowing the viral genome to enter the host cell cytoplasm. The messenger RNA vaccine strategy is engineered to generate the spike protein in a stable, pre-fusion conformation. This stabilization prevents the natural conformational shift, effectively blocking the virus's critical entry mechanism. The vaccine utilizes synthetic messenger RNA encoding the stabilized pre-fusion spike protein. This messenger RNA sequence is meticulously optimized using codon usage bias to maximize expression efficiency within human cells. A fundamental modification involves replacing all uridine nucleotides with N1-methyl pseudouridine. This substitution significantly reduces recognition by innate immune sensors, specifically toll-like receptor 7 and protein kinase R. Reduced immune recognition enhances both the messenger RNA's stability and its translation efficiency. Structurally, the transcript requires a 5' cap structure and a 3' poly A tail. These essential elements protect the messenger RNA from degradation by cellular ribonucleases and facilitate efficient binding to the host ribosome machinery. The fragile messenger RNA molecule requires protection and precise delivery, achieved by the lipid nanoparticle system, or LNP. LNPs are complex structures composed of 4 distinct lipid classes: ionizable cationic lipids, helper phospholipids, cholesterol, and polyethylene glycolated lipids. The ionizable lipids are positively charged only at an acidic pH, which is crucial for encapsulating the negatively charged messenger RNA during the manufacturing process. Following injection, the LNP is internalized by host cells primarily through receptor-mediated endocytosis. Once inside the cell, the LNP resides within an endosome, an acidic vesicle. This acidic environment protonates the ionizable lipids, causing the LNP membrane to destabilize. This destabilization facilitates the crucial endosomal escape mechanism, successfully releasing the messenger RNA into the host cell cytosol. Once released into the cytosol, the synthetic messenger RNA immediately binds to host ribosomes. The ribosome translates the messenger RNA sequence into the spike protein polypeptide chain. The messenger RNA includes a signal peptide sequence which directs the ribosome to the rough endoplasmic reticulum. The nascent protein enters the lumen of the rough endoplasmic reticulum where essential post-translational modifications occur, including N-linked glycosylation. The protein then folds into its functional trimeric pre-fusion conformation. This specific conformation is stabilized by engineered proline substitutions which are incorporated into the sequence to prevent the protein's premature transition into the inactive post-fusion state. The synthesized spike protein is processed through 2 distinct pathways for subsequent immune recognition. Spike protein fragments remaining in the cytosol are degraded by the proteasome complex. The resulting short peptides are then loaded onto Major Histocompatibility Complex Class I molecules, or MHC1. These MHC1 complexes are presented on the cell surface, leading to the activation of cytotoxic CD8-positive T lymphocytes. Separately, extracellular spike protein, or protein released from apoptotic cells, is taken up by professional antigen-presenting cells. These cells process the protein and load the resulting peptides onto Major Histocompatibility Class II molecules, or MHC2. MHC2 presentation activates CD4-positive helper T cells, which are essential for coordinating the overall immune response. Activated CD4-positive helper T cells provide crucial co-stimulatory signals necessary for B cell maturation. B cells recognize the spike antigen and subsequently differentiate into specialized plasma cells. Plasma cells function as factories, secreting large quantities of neutralizing antibodies, primarily immunoglobulin G and immunoglobulin A. These antibodies bind specifically to the receptor binding domain of the spike protein. Antibody binding physically blocks the interaction between the receptor binding domain and the ACE2 receptor on the host cell. This mechanism effectively neutralizes the virus, preventing infection. This robust humoral response, combined with the cellular response from CD8-positive T cells, establishes long-lasting immunological memory. The synthetic messenger RNA is inherently non-replicating and highly labile. Cellular ribonucleases, known as RNases, are ubiquitous enzymes in the cytoplasm that rapidly degrade the messenger RNA transcript. This degradation process typically occurs within hours to days following successful protein translation. The transient nature of the messenger RNA ensures that spike protein production is strictly limited and self-terminating. Crucially, all vaccine components remain confined to the cytoplasm and do not enter the cell nucleus at any point. Therefore, there is zero possibility of integration into the host cell genome. The messenger RNA platform extends significantly beyond infectious disease prophylaxis into powerful therapeutic applications. Personalized cancer vaccines utilize messenger RNA encoding tumor-specific neoantigens. This approach stimulates highly targeted cytotoxic T cell responses specifically against malignant cells. Therapeutic messenger RNA can also encode functional proteins to treat various genetic disorders, such as specific enzyme deficiencies or cystic fibrosis. Furthermore, the lipid nanoparticle delivery technology is being adapted for the precise delivery of advanced gene editing tools, including the essential components of the CRISPR-Cas system, rapidly advancing molecular biology and gene therapy research.